Welcome to part 3 of our network biology mini course or presentation. I was fortunate to follow Donna that gave such a nice introductory talk into networks and how you characterize them and the different types of networks. And in my 2 talks, I'm going to focus on, you would say, a very specific or algorithmic angle of networks, which is identifying modules. I'll talk about 2 different aspects of this problem of identifying modules. I'll start with the basic problem, then I'll dive into network alignment-based identification of modules in my second hour. But I would claim that even though this problem seems sort of narrow maybe, when you think about networks, it's fundamental. And it's fundamental because when you start with a network, it looks like that. So you can't really make anything out of it. And in fact, what we are going to try to do is to identify the structure in this network and to simplify the network. And this is all about module identification. So let me start by defining what a module is. And in fact, this term, as far as I know, was coined by Hartwell et al. In their paper. They termed a functional module to be a set of genes or proteins that perform some distinct function. So as Donna nicely exemplified in protein-protein interaction networks, the 2 modules that we mostly think about are protein complexes, which form some assembly in the cell or some machinery in the cell that does some specific function, or a signaling pathway whose job is to convey some signal either within the cell or from the membrane to the nucleus of the cell, etc. So these would be the 2 prime examples of protein-protein interaction modules that I will focus throughout my talk. And of course, the problem with this definition of a module is that it doesn't tell you how to find it. It's something very maybe biological. So I'll show you another definition of a module which I use, which is sort of a data-driven one. And my definition of a module is simply a set of genes that behave coherently with respect to some biological property. And this sort of this coherent behavior is what will let us try to identify those modules. Of course, we will try to identify them based on some behavior that we think is important. And then, as Ron loves to say, the proof is in the pudding. We will have to check this module that we identify and seeing whether indeed it performs a distinct biological function, etc. But most of the time, our way of defining or looking for modules will be very empirical. We will look for a set of genes that display some coherent property. Now, when thinking about module finding, the immediate association that at least I had when preparing the talk is that of clustering. Because clustering is a very old fundamental problem. Everyone touches it in his or her life. And you would think that module finding is the same as clustering. But I'd like to give 2 specific distinctions between module finding and clustering that I think make these problems different, although you'll see in the rest of my talk that I will also mention clustering approaches to module finding. So one distinction is that modules can overlap, while in many clustering formulations, sort of the assumption or the way the objective function is phrased is that the clusters do not overlap. And it's very important in biology to recognize, at least in the context of the networks that we will work on, to recognize that a protein can have multiple functions, and therefore it makes sense to have as an algorithmic output clusters that do overlap, or modules that do overlap. The second distinction is that, well, maybe it's not a distinction because we don't really know, but it's at least not a prerequisite that we cover all our network with modules. So maybe some proteins are not part of the modules. They do function by themselves, even though we know that many proteins do not work in isolation, but rather work with some other proteins. And maybe we just don't know, or maybe the network is not detailed enough so that we see everything. So I think that good approaches to module identification need not worry about covering the entire network, and it's perfectly fine to leave some regions of the network unmoduled. Okay? And I must say that also in clustering there are variants, maybe not the classical ones, but there are variants in which we don't care about covering the entire network, and maybe we leave singletons that are not, divided into modules. So these two distinctions, actually, if you think about it, give rise to, or have a similar flavor to a problem which I happen to work on, and probably many of you, which is called biclustering. It arises mainly, it popped up in computational biology mainly when analyzing gene expression data, trying to identify sets of genes that behave coherently across a subset of the conditions, while clustering would find sets of genes that behave coherently across all conditions. So there it's exactly the same distinctions between biclustering and clustering. So in biclustering, indeed you allow your modules to overlap, and you don't care about whether they cover all your data or not. Indeed, what I will show that module identification in protein interaction networks has some similarities to biclustering, and we can actually use biclustering algorithms to improve module identification in those networks. So what we will try to do in this first hour is to talk about this machine that takes this gigantic ball of protein-protein interaction and tries to identify in it functional structures that correspond either to protein complexes or signaling pathways. And the main challenges that we will deal with are twofold. So one of them is, how do we score or model? What are we looking for? What is a module? So suppose I give you a set of genes. Is it a good module or not? We will spend some time on that. And of course, the other challenge that we will deal with is, given a scoring function, how do we now go about and find the highest scoring modules? Okay, so this will be the detection challenge. So the outline of the rest of the talk is as follows. I will describe different strategies for identifying protein complexes. I will start with local strategies that sort of work on one complex at a time. I will talk also about global strategies that try to identify all of them simultaneously. I will mention biclustering approaches to protein complex prediction. And then I will mention some work on pathway inference, although the bulk of the work that I'm aware of is on identifying protein complexes. We will talk in a second why, but it's because of its sort of similarity to clustering. And finally, I hope I will have time to go into more sort of involved algorithms that try to analyze several networks together in order to better identify protein modules. So this is the protein integration topic. And I'll start with local prediction strategies to protein complex identification. And before presenting this, let me just make a brief apology. Of course, everything I'm going to present is a bit biased by, my own view of things. I didn't have enough time in 1 hour to cover everything. I wanted still to give some algorithmic details on some of the methods that I'm going to describe. So apologies if sort of, my bias is one way or another. So what is a protein complex, or how are we going to find protein complexes? The, easiest notion, if you think about it, of what is a protein complex is a set of proteins that work together So they are probably densely interacting in order to form some machine in the cell that will function together. It may not be the case, but we have a lot of empirical evidence that protein complexes are denser than random protein sets in the network. Okay, so here's just one example. Of course, it's anecdotal, but if you measure it, they have some density that is significantly higher than the density of random sets of proteins. And of course, this now gives you an almost clear measure of how you are going to identify and score those modules. So in a very rudimentary way of defining things, we are going to look for dense sets of proteins in the network. Okay? So the modeling problem that we have to tackle here is how do we measure the density. So for example, if I see 3 proteins that are all connected to one another, but these 3 proteins, each one of them have degree 100, maybe I'm less surprised compared to seeing a triangle formed by proteins that have degree 1 or 2 or 3. Okay? So one challenge we will deal with is how we model this density, how we score a densely interacting set of proteins. And the other challenge would be to find high-scoring subgraphs. So you see that we are moving, without without noticing, we are moving from the biological world in which there are very maybe well-defined creatures that are called protein complexes to the more convenient for us graph world in which, we are now looking at heavy subgraphs, which is a well-defined structure that we can try to identify. So I'll start with one of the most highly cited works, maybe also because it was one of the first for identifying protein complexes, which is the ENCODE algorithm by Bader and Hogg. And by presenting this algorithm, I will also try to mention where I see the the challenges that we still have to tackle, and from there we will grow to present the rest of the methods. So ENCODE tries to identify protein complexes by identifying densely interacting sets of proteins. The way it works is as follows. It gives it's a very simple algorithm. It gives some weight to vertices according to their potential to be in a densely interacting set. We will talk about the exact weight in a minute. And then it does the complex prediction, and the complex prediction is sort of heuristic. You start with the highest weight vertex, and then you try to join to this vertex other vertices that are neighbors of it and have their weight at least p times the weight of this highest scoring vertex, where p is some parameter of the algorithm. And so you start building complexes. Once you cannot add vertices to an existing complex, you put it aside and you continue until you cover the entire graph. How do you score vertices, which is the only, and then there is a post-processing step that I will not talk about. So the only detail that I didn't tell you is how do you weight vertices. And the way they do it is as follows. It's an interesting weighting scheme. They focus on k-cores. Okay, so what is the k-core of a graph? The k-core of a graph is the largest subgraph with a minimal degree of k. How you find it? It's very simple. You start eliminating vertices, and you end when all vertices have now a degree at least k. So you eliminate vertex one at a time. Of course, degrees are updated as you go, and you stop when you eliminated all possible vertices. So now, another definition I will need in order to describe the weighting scheme is the density of a set. The density of the set is very similar to the clustering coefficient defined by Donna. It's simply the percent of edges in a set out of all possible vertex pairs. Okay, so the way they define the weight of a vertex is as the density of the highest k-core in the neighborhood, in the closed neighborhood of this vertex, times the k of this k-core. So first they take the neighborhood of the vertex, and they see what is the largest k that gives rise to a non-empty k-core. Once they identify this k-core, the score, the weight they give to the vertex, is k times the density of this k-core. Okay? Now this is the entire algorithm, and when you think about it, it has the right intuition. It is finding dense structures in the protein interaction network. It was widely used. It was very successful. However, the definition of a complex here seems a bit heuristic in the sense that there is no underlying probabilistic model that tells you whether this complex that you now identified, or this module that you now identified, is indeed surprisingly dense. So the definition here is not statistically based, it's more combinatorially based, right? And so this is one challenge we would want to tackle, or we would want to have some assurance for the statistical significance for the modules that we identify. And the other limitation, although I will not dwell into that, is that by definition of the algorithm, the complexes cannot overlap, even though in a post-processing step, they could be made overlapping. Okay. So I'd like to focus on the first point of having some statistical measure of density. And for that, I'd like to describe an algorithm which I was part of, which is called Network BLAST, which sort of I think its main part is about the scoring, and the other things are sort of done, and I will talk about it in a bit, are sort of done as you would, as you would think about. Everything else is sort of done in a standard way. But what is special about it is the scoring, is how you try to identify not simply dense sets of proteins, but rather statistically significant dense set of proteins. So to do that, the idea is to use a likelihood ratio scoring, which requires you to define what is your model of a protein complex and contrast it to your model of how the protein network looks like. Okay? So let's start with what is a protein complex. So sort of the easiest definition that will ensure you that your model will sort of decompose over the edges and non-edges of the network is to assume that every edge is independent of all other edges. And since you assume that the set of proteins has to be dense, you would like every one of the edges to occur with high probability. So you take some parameter, P, and you simply assume that every edge in a protein complex occurs independently with high probability P. So this is the protein complex model. But now, even if you suppose you didn't use a likelihood ratio test, but suppose you simply looked for, for protein sets that abide to this model, still you would simply be finding dense sets of proteins, and it's not clear whether they are statistically significant or not. So you have to contrast it to something else. And the something else is, what do you think is a random, how does a random network behave? Are you surprised by the fact that you saw this triangle or not? So this is, so of course Donna talked about random graph models. And this is another important reason why we need those random graph models, because we need to be able to say something about the significance of what we are finding. And for that, we must know how random looks like. So sort of the standard in the field, because of the power law behavior of the networks, etc., is to assume a random graph model in which all the edges are random, but what you maintain are the node degrees. So you can think about it, or you can think of how you create it if you just simply take the network and now rewire the edges maintaining the node degrees. So this is sort of has become the standard in the field for random models for protein networks because it's relatively easy to derive, and we can also approximate certain things in this model. For example, we can approximate very well the probability that two proteins are interacting in this model, and not very surprising, this probability is correlated to their degrees. Okay, so the higher the degrees of those proteins, the larger the probability that they are going to interact in the random network with the same node degrees. Okay? So now once we have those two models, we can actually write down a likelihood ratio test that will allow us, or if you will, a scoring function that will allow us to now go and look for the highest scoring protein complexes or highest scoring modules. So what is this likelihood ratio? So let's look at the numerator first. So the numerator is simply the probability of the set of proteins that we are observing based on the protein complex model. So the protein complex model says that each edge should occur with probability p. So we take a product over all edges of p times the product over all non-edges of 1 minus p. So this is the numerator. The denominator is more or less the same, but instead of having p inside, it has some probability that is derived from the degrees of the nodes u and v. Okay? So now we have a measuring rod. We have a clear objective that we wish to maximize, and if we do maximize it, we have found some set of proteins that is significantly different, I mean, depending on its score, significantly different than what we would expect at random. So now intentionally I didn't give all details. In fact, you can make this score a bit more involved by putting into it not simply, p for an edge and 1 minus p for a non-edge, but you can actually account for the confidence in the edges, for the reliabilities that you have for the edges. So this is one comment. The other comment is that this score is very useful in the sense that it can be efficiently computed for a graph because it decomposes over the edges and the non-edges of the graph. So you would imagine that if you take here now a log, then you have a sum over all edges of something and a sum over all non-edges of something else, and you can put those weights on the edges and non-edges of the graph, and suddenly you have something that you can efficiently compute. And now you can use your favorite heuristic to identify those highest scoring or maybe just high scoring subgraphs. What we do in Network Blast is simply use a greedy local search. It works very well, but you could also imagine doing more involved things. For example, even, of course the problem is NP-hard, the problem of identifying a heavy subgraph, but you could imagine, for example, using integer linear programming to really identify the highest scoring subgraph. I must say that, I had once someone coming to my lab that actually implemented this approach, and there wasn't a lot of difference between the heuristic, the greedy search, and the actual ILP, but of course this now depends on the complexity of the network you are applying to, etc. Okay. So now I'd like to talk about sort of a different perspective on the same problem. Yes?
So even in a random network, random graph, you would have subgraphs that are high scoring. So how do you evaluate for that? Because out of the very many subgraphs, some of them would have a relatively high score.
So supposedly you account for that exactly here. So if in a random graph, you see, if you may, of course, as you know, if you make a lot of trials at random, even at random, you have a chance of discovering something that is very high scoring. However, you account for that in the sense that in a random graph, if you look at a very large degree proteins, then you do expect them to interact, and therefore your score for those proteins will not be so large. However, if you have two proteins that have a very small degrees, and by chance all of them interacted together, then of course it's a false positive, but nevertheless the chance of it happening is very, very small.
It's very small for a given C, but C is a subgraph of your graph, right? Do I understand it well? That C is a subgraph of
Right.
the whole graph. And in that whole graph, you have exponentially many such subgraphs for which you would evaluate this L. So in one network, you already have like exponentially many possibilities where this likelihood could possibly be bigger than on a random place.
That's a good question. I'm sure there is an easy answer to it. I cannot answer online, so maybe we can take it offline. You are evaluating an exponential. Supposedly, you are not really accounting for all of them, but supposedly you are evaluating an exponential number of subgraphs. The score tries to take care of your expectation, right? Of what is the probability of seeing an edge between proteins in a subgraph. You're asking, what is really my false discovery rate? Given that I'm looking at the exponential number of subgraphs, it's a good question. I don't have an easy answer online. But let's talk. I think we share sort of one wall in our offices. Okay. So now moving to a global perspective on this problem, again, I'd like to share with you two algorithms that have been influential. One of them is sort of this idea that you can use data you can analyze data in a way that something emerges out of it. So this is Markov clustering. And the idea behind this algorithm is that when you do a random walk in a graph, you are more likely to stay within the same dense region than to cross between regions. Okay? So this is sort of the main idea behind Markov clustering. And again, Markov clustering is something very procedural. You start with the adjacency matrix of the graph. You make it stochastic. And then you do two steps iteratively. In one step, it's called expansion. You raise the matrix. So it's matrix product. You raise the matrix to some power. Let's think about it as power 2, which means if you started with the adjacency matrix, then m to the power of 2 is simply looking at two long walks in the graph. And if you now raise it again to the power 2, you're now looking at walks of length 4, etc. And the other step is inflation. It is meant to boost the probabilities that are very high and to sort of downgrade or decrease the probabilities that are very low. So what you do here, you now look at the matrix entry-wise, and you raise each entry to the power of r. Okay? So again, you can think about this r as again the number 2. So if you start with a high probability, it will remain relatively high. If you start with a low probability, it will really diminish. And what usually happens, although it's not guaranteed, so there is no proof that this process always converges, but what usually happens is that you start with this convoluted graph, and after something between 10 or tens of iterations, you arrive at something from which you can actually, so the cluster sort of emerge. Okay? So this is one approach that has been very successful in clustering biological networks or protein interaction networks. The other approach I'd like to describe is something that is more, again, cares more about, has a clear objective, which it tries to maximize, and cares more about the statistical significance of what it identifies. So this is a famous work by Mark Newman, which is based on a modularity function that he defined. And the idea behind this modularity function is that you want to identify a set of modules or a clustering in which there are many edges within groups, but it's not enough to have a lot of edges within groups. You would like it to be much more than what you would expect at random. Okay, so this modularity function has two terms. One term is the number of edges within groups, and the other term is the expectation of this number under, again, a random graph model with the same node degrees. Okay? So notice, for example, so a nice feature of this modularity function is that if your graph doesn't have a clustering, then the trivial division is, of course, that all vertices are put in one group, and then the modularity function for this trivial division is 0. So it means that you are not, you don't have to specify the number of clusters in advance. If you find some division that maximizes this modularity function, then it automatically identifies the number of clusters. If there is no clustering in your data, then the best you will get is a modularity function of 0. So now I'd like to spend some time in explaining how this computation works. Okay, so let's define ki to be the degree of node I. Let's have big M or capital M to be the sum of all degrees of vertices, or 2 times the number of edges. Let's have Aij be the adjacency matrix, so it's 1 if there is an edge in the graph and 0 otherwise. And let's have Eij to be the expected number of edges between I and j in a random degree preserving graph. And the main lemma that we need in order to really compute this function is the fact that the expected number of edges between I and j in a random degree preserving graph is sort of approximated well by the degree of the first vertex times the degree of the second vertex over the 2 times the number of edges in the graph. And I don't have time to show you that, but it's really sort of an easy observation. And once you believe in that, then you can actually write the modularity function as the sum over all pairs of vertices that are put in the same group of whether they are adjacent or not, minus the expected number of edges between them. So now let's write it in a more computable way. Okay? So the way we are going to go about it is to denote whether I and j are in the same group using an auxiliary vector. And the way the algorithm of Newman works is that it doesn't really try to maximize the modularity function, but rather it works iteratively. Each time it takes the entire graph, divides it into 2, and then divides these parts into 2, and sort of so it works sort of hierarchically. And so we will write this modularity function for the case that we are trying to divide the graph into 2 groups, and we will just try to optimize this division, this single division, and then you can imagine that the algorithm simply works recursively with this definition, and it stops whenever it reaches a subgraph in which there is no good division, in which the modularity function is 0. Okay, so how do we do it? So if we only have 2 groups, we can use an auxiliary vector that has one of 2 values, either +1, which means that you're in the positive group, or -1, which means that you're in the negative group. And it's very easy to see that if you look at the expression half times SI Sj +1, it's 1 if you're in the same group, if I and j are in the same group and 0 otherwise. And therefore, we can now write something like this. So the modularity function, just for the case that we wish to divide our graph into two pieces, is again this sum over all pairs, but now these pairs are multiplied by Si Sj plus 1 over 2, which exactly says whether they are in the same group or not. So now we need a bit of easy algebra just to see that we don't really need this one, so it cancels out. Again, I don't think it's important, so I won't go into the details, although all the details are in the slide. But what's important is that we can derive an equivalent expression for the modularity function in which you have half times the sum over all pairs I and j of aij minus the expectation ki kj over capital M times si sj. And now this expression can be now written in a matrix form very economically as S transpose BS, where B is called the modularity matrix. Okay? Why is this important? This is important because if you have this expression, then you can use sort of spectral approaches to try to optimize it. And the way it works is as follows. You recognize that B is a symmetric matrix, therefore it has n eigenvalues with associated n orthonormal eigenvectors. And now when you apply this realization that it is diagonalizable, then you can simplify the expression for the modularity function to be the sum over I now, okay, over all vertices, over all vertices or eigenvalues of the eigenvalue times some alpha I squared, where alpha I is the scalar. So you write this S. You start with S, your vector that divides your groups into +1 and -1. You can, of course, write it as a linear combination of the eigenvectors, and alpha I are just the scalars of this linear combination. Okay, so now you want to maximize this term. And in order to maximize this term, since the eigenvalues were given in a sorted sequence, you know that the highest eigenvector is beta 1, and sort of if you start with some vector with a given norm, the best for you is to maximize the weight that you give to the first eigenvector, okay? Because beta I is the maximal value that you can hope for. And so if this is what you do, if you choose S to be sort of proportional to the first eigenvector, then you are sure that you are optimizing your modularity function. However, this eigenvector may not be an integer vector. It's not necessarily +1 or -1. So as a heuristic, what Newman does in his algorithm, he simply maximizes so he chooses a vector, a +1, -1 vector that maximizes the projection over this over this first eigenvector. And it's very easy to maximize the projection because the projection is simply the scalar product, and therefore if you choose Si to be +1, if the ith component of U1 is positive and -1 if it's negative, then you are maximizing the projection, and therefore you are computing approximately the best division into 2, and now you can think that you apply it recursively in order to get a division. So now I've shown you a number of different strategies for module identification, and the question is, how do they fare on real data? So in order to show you that, and I'm just going to do something very brief, I should tell you how I evaluate the performance of these algorithms. And the way I evaluate the performance is, of course, using gold standards for what is a module. So we started by saying that a module is a set of proteins that perform some specific function, and luckily we have the gene ontology in which all genes are categorized into terms. So, a fair gold standard would be to take the Go terms, or if you will, you can do something even better than that. In Go, there is also a term which is called molecular complex, so you could go there and you can sort of zoom in only on Go complexes, or for different species, there are specialized sets of complexes that were made by curators. For example, for yeast, a lot of time you will see in papers that the MIPS complex collection is used. So these are the gold standard that you use, and then you use some measure of precision of your finding, how specific you are in your finding, and some measure of recall, how well are you capturing the world of known complexes. And to measure that, you can simply measure your overlaps with the gold standard, or you can do something that is more statistical, for example, computing the hypergeometric enrichment of your complexes or your predictions for complexes with the known complexes. So luckily for us, no, it doesn't happen all the time. There are all these approaches. Each approach tries to show that it's the best, and therefore it compares to previous approaches, but of course it's a biased comparison. Luckily for us, there was, about 5 years ago, by a colleague of us, Mona Sinch, there was a comprehensive comparison of different strategies for a module prediction. This was done in yeast. There were two types of comparison. One was on simply taking, I think it was BioGrid, so some database of protein-protein interactions. So this includes both binary interactions and also interactions that are derived from co-IP studies. And then they did a second evaluation only on binary, so only on networks that are composed of binary protein interactions that were derived from yeast to hybrid assays. And here you see a bunch of algorithms that are being evaluated. We talked about some of them. So Network BLAST is the first. Then we talked about MCL. We talked about ENCODE. And this is the algorithm of Newman. And you can see that the different algorithms sort of have the same trend over each type of data set. So for example, for the high throughput physical interactions, you can see that the best algorithms are Network BLAST and CFinder. For the Y2H studies, the best algorithm seems to be MCL. And this is over 3 types of gold standards: MIPS that I mentioned, Go biological process, and Go cellular component. So I think that the picture that comes out of it is that different algorithms are maybe better suited for different applications, although there are clear dominations. So there are algorithms that dominate others in this comparison. Okay. So the next thing I'd like to talk about very briefly is biclustering approaches to protein complex identification. And the reason I think it's important is because you really, when you tailor an algorithm to a specific biological data set, you should think what this data set represents. So of course it's very convenient to think about protein-protein interactions as binary interactions. So a graph is very natural. However, some of the data comes, as Donna explained, from co-IP studies where you actually identify not binary interactions, but you identify a set of proteins that were sort of assembled together in a specific point in time. And maybe you want to make use of that. Okay? So the way you can make use of that, if you are afraid of hypergraphs and you simply want to capture this information, you can capture it in a bipartite graph. So you make a bait-prey graph in which sort of the maybe the baits are the ones that you captured the complex with, and the preys are the ones that were floating around and were captured with them. And now you really represent the essence of your data, because really the data was about capturing this set and then capturing this set and sort of. And these are exactly the kind of graphs to which you can apply by-clustering algorithms to try to identify sets of preys that co-occur together with the same baits. So I don't have time to go into the algorithmics. I will have to skip that. However, let me just know that it looks like these approaches, when applied to co-IP data, work much better than approaches that are tailored to binary networks or binary interactions. Okay? So let's skip that. Okay. So now I talked a lot about protein complex inference. What about pathway inference? So there was less work in this domain, so I will just mention some of it. So of course, the easiest problem to think about is simply finding a simple path in a graph. You don't just want to find any path, maybe you want to find the highest scoring path, or the sort of the most reliable path. Okay, so the problem is as follows. You are given a graph. Maybe you are given some parameter k, and you want to find the heaviest path of length k in your graph. Okay? So it turns out that this very simple problem is already NP complete. So maybe there is nothing you can do about it. Unless you are willing to invest, n to the k time and simply try all combinations of n vertices that will give rise to this path. Now, the first application of such exhaustive algorithms for PPI networks that I'm aware of was done by Steffen et al. What they looked for simple paths that go from membrane proteins to transcription factors. So this is our notion of what a signaling pathway should look like. And remarkably, using this very simple approach, they could identify real signaling pathways that are known to, were previously curated. What I will be interested in this part of the talk is to show you how you can deal with this problem in a more efficient way by resorting to fixed parameter algorithms, which means that these algorithms are polynomial in the size of your input, but exponential in some parameter. In this case, the parameter is going to be k, which is the length of the path you are looking for. Okay, so of course, so the time bound will be something like, instead of n to the k, it will be something like n times 2 to the k, which is very practical for moderate values of k. Okay? To tell you a bit about the work that was done in this domain, I should first introduce the sort of the main technique that is being used in these kinds of computation, and this technique is called color coding. It was actually invented in Tel Aviv by Noga Alon, Rafi Yuster, and Uri Tzvik. And the idea behind this technique is as follows. Instead of considering all possible, groups of vertices of size k, you start by randomly coloring the graph with k colors. And now you are looking at, or you are trying to identify subsets of vertices that are colorful, that all of them have distinct colors. Okay? And if you can identify them, if you can find such a set, then you can compute over this set, you can compute very efficiently the highest scoring path with the weights that are given. Okay? So you start by randomly coloring the graph, and once you color the graph, okay, you run some dynamic programming that simply computes, so for every vertex, what is the highest coloring path that ends at this vertex and visits on the way the subset of colors S. Okay? And this could be done very easily by simply going over all neighbors of this V and asking what was the highest scoring path that ended in a neighbor of V and visited all the colors in S minus the color of V. Okay? And again, the main idea here is that instead of now considering subsets of vertices, which would cost you n to the k, you are now in your dynamic programming only considering subsets of colors that cost you, so this costs you only 2 to the k. But of course, this can only succeed if you manage to color the paths that interest you with really k distinct colors. So what is the chance of that? The chance of that is as follows. There are k factorial colorings of the paths that interest you that will give rise to k distinct colors. There are k to the k possible colorings of this path, and therefore the probability that you are succeeding, that your path is really colorful, is at least 1 over e to the k, which means that if you now do this color coding e to the k times, then in expectation there will be one time in which you will discover the path that interests you. So this is how color coding works, and again, just the bottom line is that the overall time of this algorithm is something like 2 to the some constant of k, or exactly 2 times e, all of that to the power of k, times the number of edges in your graph, and this is of course much better than n to the k. It makes it very applicable, and this is what we call a fixed parameter tractable problem, or a fixed parameter algorithm, because the exponential part is only related to the parameter and not related to the size of the input. So this was done a few years back, but you can still see here that, you know, even for paths of length 10, you can compute them. Back then it was maybe 11,000 seconds, maybe now it's 10 seconds. But the bottom line is that you couldn't do it exhaustively. You really needed those tricks in order to be, even though the networks are pretty sparse, you really needed this algorithmic trick in order to be able to process a longer path. Okay? What does it give on a, let's see, so I'm going to skip that. What does it give on real biological data? So again, a few anecdotal examples. Here is a well-known signaling pathway in yeast, a MAPK pathway. And if you just apply color coding, or this approach of finding the heaviest path as is, you start from the same membrane protein, you go to the same transcription factor. Remarkably, you almost identify the original, you know, textbook path. So it's quite remarkable. Even when you don't succeed as well, it turns out that what you find is related to the path. And oftentimes, the reason why you are not succeeding so well is because there are gaps in the network, which requires the path to go through some unrelated proteins that are there just because the real interaction was not part of the network. Okay? So I'm sorry for having to move a bit fast, but let me get now to the last point of my talk. And the last point of my talk is that you can use all these techniques now in very interesting ways when you now try to integrate information from multiple networks. And when you try to integrate information, of course, it has two important roles. One of them is sort of fight with noise or eliminate noise and incomplete data. In the other important feature that it has is that it allows you to get a more complete picture of what is really going on with the module that you're identifying, how it crosstalks with other sort of molecular events. Okay, so there are two common integration schemes that people have been using. One of them is simply to put everything together, all the information that we have together on one network, and weigh the edges accordingly, and now try to identify things that are dense in each of the networks. So for example, there is a well-known paper by Gonzalez et al. That takes this approach. The other the other way people have been trying to integrate information is by using a different model for different data types and taking all of them together in order to form something that is coherent. And I will try in the few minutes that I have left to give you one such example. Okay, so the example I'd like to talk about is a paper from the lab of Trey Heidecker about integrating protein interactions with genetic interactions. So again, a genetic interaction, or the specific genetic interaction we will talk about, happens when two genes, when you knock each one of them separately, nothing happens. When you knock out both of them, the cell dies. So this is the, it's like binary arithmetic, 1 + 1 = 0. And there are high throughput technologies that allow us to detect those interactions. And of course, the question is what we can do with these genetic interaction networks. The specific question that the study I'm going to tell you about was trying to answer is, what is the meaning of these interactions? Do these interactions happen because the cell sort of has two parallel pathways? If you knock out genes in one of them, nothing happens. If you knock out genes in the other, nothing happens. If you suddenly knock out both of them, the cell dies. This will be the between pathway interpretation of genetic interactions. Or the other possible explanation is what is called the within pathway, which means that you can think of a big complex. If you knock out a certain component of this complex, nothing happens. But now if you knock out two components, maybe the complex scrambles and it becomes essential and the cell dies. Okay? So how do we use these two very different types of information? So the idea was to look directly for these two types of explanations by using again this likelihood ratio approach, where for the within pathway interpretation, you simply look for things that are dense both on the physical network and on the genetic network. While for the between pathway interpretation, you look for things that are, you look for two subsets of proteins that each is dense on the physical network, and they have a lot of genetic interactions between them, because if these are indeed two pathways, you expect every pair of genes in those pathways to genetically interact because all these interactions eliminate both pathways. And so, they applied the scoring scheme. They got different between pathway components, within pathway components, and the way for them to evaluate which one of the explanations was more fitting to the data at hand. So they did two things. I'll briefly tell you about both of them. One of them was to use these modules in order now to predict, and this Donna alluded to, new genetic interactions. So the idea is that if you identify, for example, in the between pathway scenario, if you identify two pathways with a lot of crossing interactions, and this interaction, so these are genetic interactions, and this interaction is missing, then based on the support for this interaction, how many pairs are there, you might predict that this interaction is correct. While for the within pathway model, again, if you identify a dense complex that has a lot of genetic interactions and one is missing, so based on the support for this interaction, how many other genetic interactions are there in the complex, you would think that maybe this interaction is correct. And so they did this prediction, and these are just the results for the between pathway. And the reason I'm showing that is because, so first of all, they get, using their between pathway scenario, they get a very nice prediction accuracy of about 87% in cross-validation. But what I find really interesting about this graph is that when you don't use the physical interaction data at all, if you simply try to support an edge by these genetic interaction motifs, and you ignore the physical interaction data, then no matter how many motifs are supporting it, your performance is quite poor. However, once you you integrate the physical interaction information, then your performance accuracy can go really high. Okay, so just to wrap the story up, what they conclude is that a lot more interactions could be explained by the between pathway model compared to the within pathway model. The within pathway models were more functionally enriched compared to the between pathway models were more functionally enriched compared to the within pathway models. And finally, they could get higher accuracy in predicting genetic interactions, so 87% compared to 38% when using the within pathway models. So to summarize, I've tried to explain how I view what modules are. I said that they take different shapes. I focused on protein complex predictions, but as I tried to allude to, there are many types of modules we can try to compute. I presented different strategies for identifying them. For protein complex prediction, there were local strategies, global strategies, and bi-clustering strategies. I've shown you that by integrating different types of information, you can enhance your prediction accuracy. And finally, let me just end with a note that even though, module prediction is sort of many people have moved from pure module prediction to different areas, I think this problem pops up in many new research endeavors in the form of deriving modules from multiple information types, for example, disease modules, drug response pathways, and so on. So I think this, even though maybe it will be hard to sell a new clustering algorithm, there are new contexts in which it's very interesting to devise those algorithms, and this problem, as always, is very active and very interesting. Thank you.
Is there any question?
Also, usually in graph theory, if you are given very messy or very dense graph, people use sparsification based on some like the spectral method to reduce the redundancy, something like that. So I'm just wondering, in this area, is there anybody consider to do sparsification before like perform clustering or some strategy, as you mentioned in your talk?
Mm-hmm.
I mean, yeah, it cannot directly solve the problem, but I think sparsification can make you problem either.
I mean, if you will, even the Markov clustering that I've talked about is sort of a sparsification because it starts with a full graph and sort of a clustering emerges. But my other side of answer for that is that we believe that the networks are, of course, optimized by evolution, and therefore you don't think they are redundant in any way, and you really need all the information, and all the information you care about all the information. Even though I agree that for clustering it may be, well worthwhile to use different types of method that, process the data.
The graphs are already pretty sparse. You can apply many of these techniques. So if you're looking for dense networks, we have a bunch of papers that you may remember. Although there are no guarantees with respect to the sparsity in terms of performance. You can apply some data mining techniques that seem to work very well in practice. You just build from bottom up. But you don't.
So of course, but it really depends on the application. So, for example, we wrote a program that can identify the largest click in a graph. It works very well, but for some graphs that we are given, for example, derived from protein structures, so we work a bit on that. There, the program is not strong enough, and because the graphs are, grow bigger and become denser. So maybe, other approaches are always important. But I agree that, so most of these networks that we talked about are quite sparse.
Mm-hmm. I was just wondering, for instance, in the evaluation that you showed, how many of
Hundreds. Hundreds.
The size of the network is about
So for yeast, it's about 5,000 proteins and about, depending on which network they use, between 50,000 to 100,000 interactions.
Alright, but there are many, so in such a network there are hundreds of protein complexes. That's the number.
There are more, but in the database there are hundreds of well-curated complexes.
But many of the complexes that are real are size 3 or 4. You probably want the bigger ones. But statistically, it's not strong enough to identify click of size 3.
Yeah, no, this I mean, you had this slide with the performance.
Yeah, so there it was hundreds. It was something like 200 MIPS complexes. You know, there are, for Go terms, there are thousands or maybe tens of thousands of Go terms. So it depends on what you're looking for. But if you are just interested about curated protein complexes, there are hundreds.
All right, perhaps we can make a break, and we'll start again at 3.